Everyone knows their immune system keeps you pathogen-free, but how does it actually do it? There's three main stages of an immune response. First, there's the innate immunity, then there's the early induced innate response, and finally the adaptive response. So first we have a pathogen, and that pathogen will be contained by an anatomical barrier, and thus this prevents the infection. But if the pathogen breaches through this anatomical barrier, it will result in an infection. And during the infection, there will be recognition by preformed nonspecific and broadly specific effectors, and this will result in the removal of the infectious agent. But if this step fails, there will then be the recruitment of effector cells, and there will then be the recognition of PAMs and the activation of effector cells and inflammation. And this is the removal, and then there will be the removal of the infectious agent. And this stage is known as the early induced innate response, which happens between 4 and 96 hours. But if the early induced innate response fails, there's then the transport of the antigen to the lymphoid organs, recognition by naive B and T cells, and clonal expansion and differentiation to the effector cells. And hopefully this will then result in the removal of the infectious agent. The transportation of the antigen to the lymphoid organs is known as the adaptive immune response, and it happens late in the infectious cycle. It takes about 96 hours. But not all pathogens are given the opportunity to cause infection, and this is partly due to the epithelial surfaces, which is this step here. There's mechanical, chemical, and microbiological barriers built into these epithelial surfaces, and we'll look at the mechanical barriers first. So, for example, in the skin, there's epithelial cells that are joined together by tight junctions, preventing the bacteria or pathogens from slipping in between the cells. On the skin, there's also the longitudinal flow of air, brushing off the microbes. And for the chemical barriers on the skin, there's fatty acids, which are hydrophobic, and they prevent the bacteria from entering. And there's also beta-defensins. And beta-defensins are antimicrobial peptides that prevent microbial colonization of the epithelium. There's also things known as lamellar bodies, which secrete polymerase surfactant into the extracellular space. And there's also catalycidins. And catalycidins are antimicrobial peptides stored in the lysosomes of macrophages and other phagocytic cells. And there's also the microbiological barrier. And the microbiological barrier is just the normal microbiota of all the bacteria living on the surfaces of the skin. In the gut, the mechanical barrier are the epithelial cells, which are joined together by tight junctions. And it's also the longitudinal flow of fluids in the intestines. The chemical barriers are low pH, enzymes such as pepsin, and alpha-defensins, along with catalycidin. And the microbiological barrier is also the normal microbiota. In the lungs, the mechanical barrier are the epithelial cells joined together by tight junctions, and there's also the movement of the mucus by the cilia, which is known as the mucociliary escalator, which moves the mucus up and out. The chemical barrier is polymerase surfactant, and there's also alpha-defensins and catalycidins. And for the microbiological barrier, it's just the normal microbiota. And in the eyes, nose, and oral cavity, the mechanical barrier are the epithelial cells which are joined together by tight junctions, and there's also tears in the nasal cilia. The chemical barrier are the enzymes in the tears and saliva, which is lysozyme. And there's also histidins. And histidins are an antimicrobial protein found in the saliva, and there's also beta-defensins. And the microbiological barrier is the normal microbiota. This video is going over the first line of defense, which is the epithelial barriers. In the next video, we'll go over the stage of innate immunity.